Hello, I'm Ron Vale, and today I have the great pleasure to be speaking with Jim Rothman and Randy Schekman, who just returned to the United States from Stockholm, where they received the 2013 Nobel Prize in Physiology or Medicine, along with their colleague Tom Sudoff. This trio was awarded the prize for their seminal work in uncovering the molecular machinery that's involved in membrane trafficking and protein or neurotransmitter secretion. This is a journey of proteins through the cell that starts with protein insertion into the endoplasmic reticulum. Those proteins are then recognized as cargo, packaged, and then transported as small vesicles to the next way station, which is the Golgi apparatus. And from there, they are transported to new membrane destinations, such as the lysosome or the plasma membrane. And this trio of Sudoff, Rothman, and Schekman found the molecular machinery that's involved in each step of this pathway through the cell. So thank you for joining us today. And I'd like to begin with the science. And maybe, Randy, I could ask you if you could describe the work that Jim did that led to his contributions for the Nobel Prize.
Happy to do that. I first met Jim when he interviewed for a faculty position at Berkeley. As an assistant professor, I'd been there about a year. And he told me in broad terms about his plan, which I certainly appreciated, being a biochemist. And then he went and did it at Stanford. Very quickly, he launched into developing a cell-free reaction to reproduce traffic. At least initially, he thought it was traffic from the ER to the Golgi. Subsequently, it turned out to be measuring traffic, vesicular traffic within the Golgi. And he used that to identify soluble proteins that he called NSF, for N-ethyl maleimide sensitive protein, and then SNAP, which was the membrane receptor for NSF, and showed that these were involved at some point in membrane fusion. And then in a really brilliant experiment several years later, he used the NSF and alpha-SNAP complex bound by non-hydrolyzable ATP analog to fish out of membranes from a crude detergent-soluble fraction from bovine brain the proteins that turned out to be the membrane anchors and the likely catalyst for fusion, which he called snare proteins. So it was a really beautiful synthesis of biochemistry and functional analysis.
And Jim, maybe I could ask you the same question. Could you comment on the key discoveries and contributions that Randy made that led to his being awarded the Nobel Prize?
This is a great pleasure to have a chance to. Randy and I have been joking for a long time about giving each other seminars, so you've given us a great chance to do that, Ron. Randy, starting in the late 1970s, took on a problem that was extremely adventurous. And people at that time, model organisms were not what they are today. There were very few examples where, for example, yeast had been used to really forge ahead and understand complex eukaryotic pathways. As Randy points out, the yeast cell cycle was one. People doubted, actually, that there was a secretory pathway in yeast that reflected in any meaningful way the similarity to the Pilati pathway that we all had understood at the time. The Golgi apparatus, if it was observed, it was only scantily observed. And Randy, especially in a really innovative approach, was able to isolate a series of mutations, first based on kind of brute screening, and then after that based on a really important experimental insight about if you have a cell that is making protein but not excreting the protein, it's going to get denser. And so based on that, Randy was able to isolate a large number of mutations systematically. They essentially defined the entire secretory pathway and the major secretory organelles in yeast and established the generality of the pathway. But more importantly, they provided the means to discover the molecular machinery that is important in various stages of vesicle budding and fusion. And it's turned out over the years that the genes that Randy initially discovered, 23 of them, play pivotal roles at multiple stages of the secretory pathway, some at all stages of vesicular trafficking. His students and others have followed in his footsteps and have essentially laid out the entire map of the transport pathways in the yeast cell.
So both of you shared some interesting similarities in terms of how your careers started. You're both trained as biochemists in some great laboratories, and you then became assistant professors, both at a very young age. And then both of you picked very risky problems to start your laboratory. So I'd like to get some background of how you came up with this project and how you decided to tackle it. So maybe I can start with you, Randy. Maybe you can just tell me how you ended up choosing the problem membrane secretion and also taking a genetic approach.
Near the end of my graduate career, in looking at the literature on membranes, largely influenced by Bill Wickner, who was a postdoc in Arthur's lab at the time, who had also come from Kennedy's lab, that biological membranes were likely to be a subject that one could learn a great deal about biosynthesis. One could possibly treat a biological membrane as a macromolecule and dissect its biogenesis using the techniques of biochemistry and genetics in a way that hadn't really been applied yet. So I went to John Singer's lab to learn about what he was thinking about biological membranes, and I found myself on a project that didn't really have a future in my mind. It was using a scarce material, and I found that working with mammalian cells, at least at that time in the mid-1970s, was not as satisfying as working with a microorganism. So I resolved, when I joined the faculty at Berkeley, influenced to some large extent, I would say, by the success of Lee Hartwell in his genetic dissection of the cell division cycle, that I would then launch an independent career using some kind of genetic analysis of membrane biogenesis. And Peter Novick was one of my first graduate students, and it was my good fortune that he joined my lab. Very thoughtful, very precise, hardworking, really brilliant student. And so the two of us sort of embarked on this effort to identify the genes involved in secretion by a genetic approach. It was a kind of a trial and error. Initially, we had some crazy idea about the need for a genetic selection. That didn't work. We found a mutant in spite of it being a selection that didn't work. We found the first mutant anyway. And we realized that it was a complex pathway and that probably just looking through a random assortment of temperature-sensitive mutants, just as Hartwell had done, would turn up more genes. And we did find another one, but then, as Jim mentioned earlier, we decided to use an enrichment procedure and isolate a lot more mutations. So it was a good choice, obviously. It worked out well. It's not the traditional way that one starts a career. Usually postdocs are a little more conservative, but I think I always advise my postdocs to be more bold and to do something new. But of course, when you do something new, there's a good chance that you'll fail. So what I did was I started several different projects. This one worked within a year, and then I marshaled all of my efforts to focus on that.
So as a follow-up, I was wondering if you could explain the assay that you used to get the Sec mutant, and also your reaction to that amazing electron micrograph in that figure 6 of your 1979 paper that shows this yeast cell filled with all of these vesicles that could no longer reach the cell surface.
So the idea was that in looking at thin sections of yeast cells, one can see some organelles that look like they could be associated with secretion, not as dramatic as in a section of mammalian cells. But Novick and I guessed that the vesicles that one sees congregating under the bud portion of a dividing cell would be the vehicle not only for secretion, but for assembly of the plasma membrane. And so we predicted that anything that blocked the biogenesis of these vesicles would be lethal, and therefore one would have to go after conditional, temperature-sensitive lethal mutations. So that was the first and crucial assumption, that it was going to be an essential gene. And the second was that the mutant would, at a restrictive temperature, cause secretory proteins, which are otherwise very efficiently secreted from cells, to be accumulated inside of the cell. And that actually is a really trivial assay. You just assay intact cells, which measures the enzyme that's been trapped in the cell wall, because the cell wall is porous. And then you dissolve the cell wall with an enzyme, and you have spheroplasts that are left, and you just rupture the spheroplast with a detergent, and you assay what's inside the spheroplast, and that's a measure of accumulation. In a wild-type cell, that baseline is virtually zero. In a sec mutant, it rises dramatically above the background. So the first mutant, just based on a biochemical enzyme assay, was really obviously blocked in multiple secretory enzyme transport. But the dramatic result came just around the time George Pilati visited Berkeley for a special lecture. And I recall Novick telling me that at a luncheon, Pilati had encouraged him with these preliminary results to cut a thin section and look and see what these cells look like. And so we had an expert technical assistant in the department. And that, of course, was one of the great eureka moments of my career when Novick excitedly called me down to the EM room and showed me this Sec1 cell sections that were just, like they had measles. They were just full of vesicles. And nothing like that had ever, at least never been published. I don't think it had ever been seen before. And from that moment, literally from that moment on, I knew that we would have at least several decades worth of productive work.
Well, Jim, let me ask you a similar question. You started off your career at Stanford as a very young assistant professor. You had experience with the VSV virus from your previous work, but you decided to tackle a very difficult problem, which was to try to reconstitute parts of the protein secretion pathway in a cell-free extract. And in fact, many people thought that wouldn't work and that it would be an extremely risky project to take on. So I was wondering if you could give a little bit about your thinking and the background that led to that particular project.
My exposure to this entire area of biology began as a medical student in my first year at Harvard Medical School. And George Pilati's discoveries were really fresh, and they were presented in my histology course. And when I saw those electron micrographs of vesicles and endoplasmic reticulum and Golgi and so on, I immediately started thinking, given my background in physical sciences, how in the world could such a vesicle form? What would make that happen? How would it know where to fuse? And I must say, somehow I identified that problem as one that I wanted to solve at some point. And I got fixated on it. And I can't really tell you why, but I just got fixated. I found it as fascinating then as I do now. And after leaving medical school and finishing my PhD with Gene Kennedy, where I did learn some basic biochemistry of membranes and lipids, I went to work with Harvey Lodish for a year or two, and I did it with the freedom of having my assistant professor offer in hand. So I was able to do pretty much what I want and focus on really educating myself. And from Harvey and his colleagues, I learned how to work with complex cell-free systems. We were using cell-free extracts that were synthesizing protein. We learned, following the footsteps of Günter Blobel, how to study the incorporation of those proteins in vitro into the endoplasmic reticulum membrane. And that really equipped me very well. Now, when I went to Stanford in 1978, like Randy, I pursued several different projects. And that was a way of distributing the risk. But then again, I made a decision to focus intensively on one project when we did have some success after some period of time, and that's the project you're referring to, the reconstitution of transport in a cell-free extract. And I think that decision that Randy made and the decision that I made is a very important decision that young scientists can make, which is at some point you really have to focus, and you have to focus deeply on something. And in the long run, if you want to make a contribution that's really worth making, rather than just simply incremental, you have to stick, as Randy did, and frankly as you did, Ron, to that decision. It's not always going to be easy. It's certainly not free of risk, but it has great rewards for science when you're successful. And so what we did was we wanted to reconstitute transport from the ER to the Golgi. So we took the cells living. We gave them a very short pulse label with radioactive methionine. And, tricks I'd learned in Harvey Lodish's lab, then homogenized the cells to retain them in the G protein in the endoplasmic reticulum. And then we incubated that extract to see if the glycoprotein could be further processed by adding certain sugar residues, especially N-acetylglucosamine, which we knew from the work of Phil Robbins and Stuart Kornfeld and others occurred in the Golgi. So if we could see that processing in vitro, it would have suggested that transport occurred. And indeed, after, I don't know, a month or two, we observed that was the case. We could see it. But that we had a huge background, even without any kind of an incubation of longer than a few seconds, even without adding ATP or cytosol, we still saw processing. And what we came to realize was that we already had too much of the G protein sitting in the Golgi, and it was getting glycosylated directly there. So that gave me the idea of what is now known as a complementation assay, which is pretty much the standard design for in those days, most cell-free reconstitutions of transport. And what the idea is, you want to mix two extracts, one from a cell that contains the cargo protein, VSVG, but lacks the processing enzyme. The other extract from a cell that lacks the processing enzyme, but, has the processing enzyme, but lacks the cargo protein. And so when the two are mixed, if the cargo protein is getting glycosylated, it must have gone from a donor membrane in the extract of the one cell to the processing enzyme in the Golgi of the other cell. And so that eliminated the background that I referred to, because the extract with the VSVG protein was incapable of processing by adding sugar. So when we set that assay up, it actually worked. It was in, I believe, 1979. I still have the gel. It shows a very, very faint band of the VSVG protein being processed to what's called an N2H-resistant form. It was very, very exciting.
Randy, you were originally trained as a biochemist with one of the master biochemists, Arthur Kornberg. You then began to pursue this problem of membrane secretion in yeast using a genetic approach. When did you decide it was the right moment to start to begin a biochemical dissection of this problem?
Our approach to initiate with genetics was always, in my mind, just a stepping stone into biochemistry. I didn't want to pursue it purely by genetic analysis. I was every intention of using this to develop some kind of a cell-free reaction. It was a challenge to get people in my lab to think that way, because when something's working, everybody who joins your lab wants to keep doing that thing. You know, they wanted to isolate more mutants. That was great. They wanted to clone genes, but convincing any of them to actually take a step back and do something very different, which to me would have been the obvious thing, was tough. So fortunately, after about 6 or 7 years, David Baker joined the lab, and he was brash and considered our efforts in that direction inefficient, ineffective, and within 2 weeks, he had developed a cell-free reaction that reproduced the first half of the secretory pathway in vitro. And from that point on, everyone who joined the lab said, okay, I want to do the biochemistry, because it was obvious how valuable that would be. So what's the lesson here? I guess, you have to have the courage of your own convictions to be persistent, and I think particularly this kind of a biochemical approach, if you've had success in that in the past, you have, as an article of faith, this really certainty that you can do it again. And I felt confident that we could, and of course Jim had succeeded, so I felt doubly confident that we could do it. And but that was a plan right from the start.
Jim, I know you were originally trained as a physicist. Then you did beautiful physical chemistry work with Eugene Kennedy. And eventually you managed to mature the research problem in vesicular trafficking to the point where you were able to get purified membranes and purified proteins, the snare proteins, and reconstitute a membrane fusion reaction. So that must have been a very satisfying moment, and I was wondering if you can explain how you got to that particular transition where you could study this problem in this really well-defined manner.
The history of that work was we were left logically with NSF, SNAP, and snares as the components of the fusion machinery. Bill Wickner did an experiment that in his vacuole fusion system that established actually that NSF does not participate directly, and NSF and SNAPs do not directly participate in the fusion reaction per se, when put together with our earlier finding that the ATP hydrolysis by NSF disrupts the snare complex. He essentially educated us, somewhat reluctantly, I might add, that NSF must be, while it's critical to energize the system, is acting either after the fusion reaction or prior to separate a pre-existing snare complex. In any case, it left the snares, pardon me, as the only logical candidate. So with that in mind, I would say we weren't surprised that the snare complex mediated fusion. And later that year, Reinhard Jahn and Axel Brunger had published the X-ray structure, which showed a hairpin arrangement. And so it all really came together, in that respect in 1998. And you might say since then we've been trying to study more or less the physical chemistry. My current focus is the still unresolved problem of exactly how calcium, synaptotagmin, and complexin put a brake on fusion at the last millisecond in the case of the synapse, and how they orchestrate in a highly amplified and synchronous fashion the release of the neurotransmitters. That also is a problem in physical biochemistry, which I find enormously satisfying. So it's sort of fun for me at the end, towards the end of a relatively long career. It's been 40 years almost in research since I left university, to come back and have an opportunity to really take this problem to the structural and biochemical level.
So I'm curious when you first became aware of each other's work, and also while all these exciting developments were happening in each of your own labs, were the two of you in communication with one another?
We were in touch with each other. I mean, after all, Jim was at Stanford, I was at Berkeley. We actually had 1 or 2 day-long joint group meetings.
2.
Was it 2? Yeah.
Yeah.
There was some tension between us at the time. I think it was because, we both wanted to make our name, and and it was a new area, and we were carving out our own approach, and that was.
But we still were very much in communication,
yeah.
and it was very open.
Yeah.
And I'll tell you something that's a little different today. Oftentimes, I mean, there was never a time that you didn't share an idea with me that you were thinking about.
Yeah, we weren't doing exactly the same experiments.
And I always felt comfortable talking to you about something that I was thinking about, even before we started the experiment. Mm-hmm. Because, it's just there's a certain pride of craftsmanship and respect that really good colleagues have, even if they are competitive with each other. Yeah. I think we always had that.
Yeah, it was, but there was still tension, and I think that dissolved later on, especially when it became clear that we had identified genes that were encoding the proteins that he was studying. At that point, actually, I think we both felt that we could we could establish ourselves at the same time, but through a different approach.
So obviously both of you have had incredibly exciting careers in the field of membrane trafficking. If you had a chance to do it all over again right now, would this be the scientific problem that you would pursue?
Jim and I have had this conversation, which is, if we were young again, would we start in this field all over again, and in 2013? And the answer, for very personal reasons, is no. I would not, and I think Jim shares this feeling.
No, I wouldn't either.
Because it's a relatively mature field. I think there are many interesting things to discover. But Judd, you heard what I said earlier. I wanted to make my own identity. I didn't want to be in someone else's shadow, and so for that reason, if I were starting over again, I would pick something brand new that was underexplored, and where I'd have a lot of time and freedom to develop a novel approach and make new discoveries, really significant new discoveries. And so I don't mean to discourage people from thinking that way, but
if
I
I
think...
could say at another level, I totally agree with what you said, Randy. But it all depends on your scientific focus. So if I was I'm a professor of chemistry at Yale. I'm getting a lot of interest in my work now from the chemists. Because at another level, this is now a problem where the biology is considered relatively solved. Nothing is ever completely solved. Nobel Prizes aren't given out for stuff that isn't basically solved. On the other hand, it's a problem that's just been defined from the point of view of the physical biochemistry and the chemical biology that can come from it. So depending on your worldview, for cell biologists or developmental biologists, there's probably other things to do that are really bigger challenges today. For chemists, physical chemists, even physicists and people interested in modeling, it's a really, really prime area.